Good morning, everyone. The webinar is now live and being recorded. As everyone joins in, we'll start the webinar in a few minutes. We still have some people checking in, so we're going to give them like 1 to 2 minutes, and then we'll start the webinar. If you have any questions, please feel free to type them into the chat bar, and we'll get those questions answered for you. And we'll review some other things about asking questions during the webinar as well in a few minutes. Again, thank you for attending. We're just going to give people a few minutes to log into the webinar. Hello, everyone, and good morning. We were just giving people a few more minutes to check in, or a minute or two to check in. Still have some people joining us, so we're going to wait like 1, maybe 2 minutes until people stop joining us, and then we will start the webinar. So if you'll hold on for that time. In the meantime, you can enable closed captions today by clicking on the caption button in your Zoom window, and the show captions button. Again, we'll just give people probably 1 more minute and then we'll get started. Hello, everyone. Thank you for your patience while we let our attendees check in. It looks like we have a good group, so we're going to go ahead and get started. If you're new to our webinar series, welcome and thank you for joining us. Criterion Edge runs multiple informative webinars throughout the year. Today, we're presenting a webinar in our Ask the Experts series entitled How Much Data is Sufficient? Navigating Clinical Evidence Requirements under MDR and IVDR. Sarah, go ahead and go to the next slide, please. All right, before we begin, I'd like to mention a few housekeeping items. If you experience any technical difficulties, please use the chat option in your Zoom menu bar to send us a message, and we'll do our best to help you resolve it. You can enable audio transcription for our webinar today by clicking the captions button and clicking show captions. A full transcript can also be enabled so that you can follow along with our presenters throughout the presentation. If you have any questions about our content today, you don't have to wait until the end. There is a Q&A option on your Zoom menu bar. Send in your questions at any point during the presentation, and we will run through as many as we can during this session. If we don't get to your question, we will get in touch with you later by email, so it won't go unanswered. Throughout the webinar, we'll also be sharing important resources and links in the chat for our attendees to access and other related webinars or information sources. This webinar is being recorded and will be available to watch on our website afterwards. You will also receive an email with a link to the recording and the slides. Lastly, I encourage everyone here to take our short survey once the webinar ends. Your feedback helps us create content that matters to you. We're glad you joined us for the webinar today with Sarah, and they'll be answering questions on navigating clinical evidence requirements under the MDR and IVDR. So first, I'd like to present our presenter for today, who is Dr. Sarah Chavez. Dr. Chavez is the Director of IVD and Scientific Writing Services at Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of lab bench experience. She has taught at the university level for almost 20 years. She has extensive scientific writing experience in academic environment, as well as several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. Now I'm going to go ahead and hand this back to Sarah, who'd like to present some information, a little bit different for our Ask the Experts. Sarah wanted to present some information that she thought would be helpful before getting into your questions. So with that, I'll hand it back to you, Sarah.
Okay, thanks, Mark. So as Mark said, we've come across similar questions in each of our Ask the Experts webinars over the years. And it really comes down to this concept of sufficient clinical evidence, because both the MDR and the IVDR refer to this concept of sufficiency, but it's very subjective. How do you know what is actually sufficient, and how can you set out a process where you're confident that you have developed enough evidence to actually meet their expectations? So that's really the background behind today's webinar. We wanted to discuss this concept of data sufficiency and use some real-world examples from both the MDR and the IVDR space to really help you understand what the expectation is. So we're hoping that through presenting examples of both legacy and novel devices, we can give you some tips as to where to actually look for the evidence, but really just to describe that process overall. So first off, as we're getting started and we're starting to receive some additional questions that you guys can feel free to send in during the course of this Ask the Experts, we wanted to start with some definitions. So when you look up clinical data in both the MDR and the IVDR, there are some consistencies. In all cases, we think of a clinical investigation. So that's pretty typical. But you can also have that peer-reviewed scientific literature is one aspect. Those are going to be things like consensus expert opinions or positions from professional associations. There can also be other relevant clinical data. And this is the part that I think tricks people up a little bit, because these are the things pertaining to safety, scientific validity, clinical performance, and clinical benefits, as well as the design characteristics and the intended purpose of similar devices. IVDR really does a good job of setting those out. Those aren't really called out in the MDR definition. They're more or less implied. So keep that in mind. Data is not just a clinical study that you conduct on your particular device. There are many sources of data. So one of the questions that we received in the registration for today's webinar was on data sufficiency. Someone asked if we could expound upon not only how much data is sufficient, but also how do they factor in the quality and the level of clinical evidence? And ultimately, that's a really good question. We wanted to point out that the sufficiency describes whether or not you can support your intended use. Can you support all indications, all target groups or populations? Are you supporting every one of the clinical claims that you are presenting? Have you described any risks or any of the analytical and clinical performance features and performance objectives that have been documented in the literature? And have you taken into account all of the relevant characteristics as you're generating the data here? Are you, doing that deep dive into making sure that your competitors are the closest thing to your device? And then also, describing the body of scientific evidence that you're pulling from. Are you a novel device? Are you a legacy device? Are there a lot of different options in this particular either treatment space or diagnostic space, or are you it? All of those come into play when you're describing sufficiency. But then when you're looking at the quality, that's really where you start to take into account, some of the conflicts of interest. Where are these studies being published? What are the study designs? Are they appropriate? Is your data being reported sort of as not the focus of that particular study, but just sort of presented as a side note? You really want to take into account the state of the art. And this is where we want to take into account appraisal of the data. That's going to really help you drill down to not only do I have enough data, but is the data that I'm presenting sufficient to make the argument here? So taking a step back, when, we've been describing clinical data, but ultimately clinical evidence is what the MDR and the IVDR are requiring. So what's the difference? A lot of times people will use those terms interchangeably. And this is a really critical point that the notified bodies have made at recent RAPS meetings. When you're talking about clinical evidence, you're not only describing the data, but you're describing your clinical evaluation or your performance evaluation if you're an IVD. And what that ultimately means is that you're not just presenting the data, but you're presenting your analysis and your summation of that data. Don't expect the notified body and the reviewers to make that jump. Just because you present all of this data and you've got it laid out nicely in tables, if you don't explain what that data means and how you are state of the art, that's what they're expecting. So that's a really critical part of the whole evaluation process. And it also speaks to the importance of making sure that you have this data organized well across the different documents and sections, and that you also have consistency across those sections. And that can be really tricky, but that comes down to this overall evaluation process that they're seeking from us all. Okay, so I thought it would be really helpful before we start this discussion to take a step back and just describe the overall methodology that we're all undertaking. And I've tried to keep it consistent between both MDR and IVDR. So this whole process will be analogous whether you have a medical device or an IVD. So first off, you're going to start with the state of the art. And this must, in order to do a proper evaluation, you have to consider the benefit risk ratio in light of the state of the art. And that's related to the practice of medicine for the purpose of diagnosis or treatment or patient management. So you're ultimately trying to establish that there's a sound scientific principle that is driving the use of your device in question. So you're asking yourself from a state-of-the-art perspective, are you compliant with the latest international standards? Are you actually the correct analyte or the correct technique or technology to be used for that particular condition? If you're talking about an IVD, is the measuring range or the test calculation considered to be state-of-the-art? And really, are you compatible with other or comparable to other devices on the market, or are you recommended by current clinical guidelines? So all of these contribute to this concept of state of the art. So what data are they looking for here? Because this is one bucket of data. So when you're talking about state of the art data, look for existing data sources. These are going to be technical standards, professional medical society guidelines, systematic scientific literature reviews. Those are a really good source. So like Cochrane reviews and things like that. And also clinical investigations or clinical performance studies. So look for any and all sources of existing published data. But don't forget, you have the option of generating new evidence. So if you're trying to establish state of the art, and there is not a whole lot out there because you're a novel biomarker or a novel treatment option, that's where the information you're gathering through preclinical studies or your own clinical investigations can be included in your analysis as sort of this new source of evidence. But that's part of where that evaluation and that description is really key to walking the reviewer through your argument, walking them through how you've arrived at the conclusion that your device is in fact state of the art. So the next step really then is trying to appraise and analyze all of the data that you've come up with. As you've searched across all these various data sources, can you say conclusively that you've made that clinical association or that scientific validity? Are they established now? And really, like, let's say you get to the end and your state-of-the-art search doesn't make that very clear. Maybe you are a device that's been on the market for 50 plus years, and now you're saying, well, am I still state of the art for all of the indications I'm claiming? Maybe your place in the market has narrowed since the time that you originally devised your intended purpose. If that's the case, and you can no longer support that you are applicable to all of the intended purposes that you're claiming, you may want to take a step back and refine your intended purpose and go back to what does the state of the art actually support. So like, for instance, let's say you're looking at something like, a pregnancy test. A pregnancy test is used to diagnose pregnancy, but that same analyte, HCG, could also potentially be used to diagnose cancer and certain precancerous conditions. If your test is claiming that you can do both and you are a urine-based assay, realistically, if they're ordering that test for the purpose of diagnosing cancer, would they really use a urine assay? Probably not. You're not going to find a lot of data out there supporting that. They almost always go to a blood test for that designation. So your intended purpose could get more narrow. It's not just what your device could be used for, but what do you have data supporting its use for? Another example here is for staples. You know, if you are like a medical device, like a staple, and you say, well, yeah, you can be used for gastric bypass surgery, and you can be used to staple the stomach, and you know, you can also be used for colon resection, but you don't have data on how well does this particular staple perform in a colon resection. You only have data supporting gastric bypass. You may want to take a step back and say, am I state of the art for all of my indications? Can I be used in pediatric populations? Can I be used in adult populations? And does the data support all of the indications and the populations that I'm claiming? That's really where this gap analysis comes in and where it's so helpful. But let's say you got lucky and you did have plenty of data to support your intended purpose. Your intended purpose is solid. That's where you then take a step of actually generating this data set, not only supporting the state of the art, where do you fit in the grand scheme of things, but then you also want to appraise and analyze that technical data. And that, if you're talking about the IVDR space, we call that the analytical performance. But from a medical device perspective, it's typically part of that technical specifications, and that can go into, you know, the metal that's being used in the device, or the alloys, you know, that can be the size, or the, you know, the particular technical features of your device. And then you're going to then say, okay, now that I've supported the technical or the analytical performance, can I then identify sufficient clinical performance data? And I think that's what we all think about when we're thinking of this sort of evidence. That's really where people get tricked up, is do we have enough data to really support our clinical performance once the device gets out of our hands and it's now in the real world? And that's that elusive data set that we're all hoping to find. So, okay, so let's suppose now you've got your clinical, you're on to that step, you're looking for clinical data. The first thing to ask yourself is, do I need to perform a clinical investigation? And I know everyone is trying to avoid that because that can be very time consuming and very costly. So first off, ask yourself, are these clinical data required? Is it a necessity? And if it is, you can then look into whether or not those studies have been performed already, or if you need to perform an additional clinical study for your device. And then at the end of that whole process, you're going to ultimately ask yourself, are the clinical performance data taken in conjunction with that analytical and the scientific validity data sufficient to demonstrate conformity? It's the grand sum total of all of these pieces of evidence. It's not one over the other. They all have to play off of each other and be part of this consistent data set. But let's say you start off and your argument is, no, clinical investigations are not necessary. Maybe I'm a very simple device. Maybe I am a staple, or I'm something very straightforward that no one questions the scientific validity. No one questions the technical specifications. And when you start digging, there's not a whole heck of a lot of clinical data out there. If you are asking yourself, do I really need clinical data? The answer is always yes. That's always going to be part of it. But do I need additional clinical data beyond what's in the literature that I've identified? Have I found those relevant data supporting the state of the art? Have I really done that deep dive? That's where, you know, if you can state that you are conforming with the relevant GSPRs, you may not need additional studies in order to make that argument. So take into account the grand sum total, because ultimately, if you can't say that after you've conducted your own clinical investigations, you're going back to the drawing board. You're going to have to say, no, actually, I do need to conduct a clinical study. If you are stating that you can be used for gastric bypass surgery and colon resection, but the data in the literature and the guidelines and all of that, and even the technical performance data is not published and is not well established, you may need clinical studies in order to expand upon your intended purpose. This can come into play even with, like, say, COVID testing. Early on, the clinical investigations that were conducted were done very quickly in a relatively short amount of time. We didn't know firsthand if this test would be reliable in adult versus pediatric populations, or in nasal swabs versus saliva. All of those things were unknowns. And so now that we're going back, your initial submission may have stated I can be used for any of those populations. But now, unless you have data supporting that the test works in a pediatric versus adult, and timing-based data, does it work early in infection versus later in infection? Those are the types of clinical investigation or studies that may be part of that gap that you'll have to undertake. And so only after you can demonstrate conformity, that's when you are ready to go, and you can actually start preparing your CER or PER. So I thought I would end with another question here that came in from some of the pre-webinar submissions. There were a lot of questions about this concept of legacy versus well-established. Where do I fit in? And how much evidence do I need if I'm just a simple lab device, or I'm a novel device? This is a very helpful piece of evidence that actually one of the notified bodies referenced at one of the recent meetings as being just a really good data source. So I highly encourage you to check out MedTech Europe. They have compiled a lot of the guidance that are out there and put them into very easy sort of flow charts and graphics like this one. But ultimately, if you look at things like these, the level of evidence that is required for your clinical performance data, it's going to drive back to that intended purpose. Get your intended purpose on sound footing before you embark on this whole process. You may not be able to support a very broad intended purpose. Your focus may need to narrow based on what is state of the art and what data is available to you, and whether or not you're willing to perform additional clinical studies. You also want to look into which group do you fall under based on those global harmonization task force designations of being either an established and standardized, established and non-standardized, or a novel device. That's really going to be the sticking point, because for novel devices, they are expecting clinical performance studies up here in the yellow. So they're expecting prospective studies, and they're also going to expect some of these retrospective or biobank leftover sample studies. If you're a non-standardized test, you may have a combination of all of those. If you're established and standardized, certainly by that point, you would expect to see all types of data, all levels of evidence. So it's going to come down to how robust is the literature search that you're performing, and have you really exhausted all sources of data? So that's what we're hoping to provide everyone with, is sort of this overview of what are the expectations of the notified body? How much evidence are they expecting you to have, and how much evidence is required based on the current guidances that we have? So here, I'm really hoping this helps provide us that context as we're moving into the additional questions and answer portion of today's webinar. I tried to incorporate some of the questions into some of these background slides just to help us sort of frame our conversation today. So a big thank you to everyone who submitted their questions ahead of time. If you didn't have a chance to, or if you've thought of something new, feel free to submit your questions now, and we can also add some additional questions at the end. So we're going to move into the Q&A portion of today's webinar, and we're excited to actually have our company president, Lori Mitchell, join us in on these discussions to help us with fielding some of the questions. Lori is the founder and president of Criterion Edge. And she has over 20 years experience in medical writing, safety and pharmacovigilance management, and regulatory reporting. And having provided regulatory and medical writing solutions to many different pharma and medical device companies, both large and small, she's been a proven leader in designing these strategies to meet these regulatory challenges. She's also a published author and holds a Master of Nursing from UCLA. So thank you so much, Lori, for joining us and helping us field some of these medical device questions that are coming in.
Yeah, you bet, Sarah, and thanks for that that very comprehensive overview. As we jump into the questions now, I'm sure the audience can tell that really Sarah's expertise lies in vitro devices. That is her field. So all questions with regards to IVD will certainly go to Sarah, because I'm more clinically based and more focused on medical device questions. So let's take the first question.
All right. So thank you guys. Thank you, Sarah and Lori. So we're going to move into questions now. We'll start with some questions that were submitted during registration, but we'll take your live questions later on as we go through the webinar. So please submit them to the Q&A button in the Zoom webinar controls that you have. And of course, if you have any issues finding this or with anything else in the webinar, just go ahead and chat and we'll help you. So with that, we're going to go to the first question. How do we evaluate how much clinical evidence is enough to support a clinical claim? Lori, I think you might want to start with this one.
Sure, yeah, I can. Sarah, for the audience, Sarah has given a really great overview and even a little bit of a deep dive on factors that really go into answering this question. How do we evaluate? And maybe what we really mean is, how ultimately will the notified bodies evaluate how much clinical evidence is enough to support a claim? And there is a one-to-one relationship in the CER, the PER, the whatever it is that the document that you're going to be writing under either IVDR or MDR. The notified bodies will want to see a very clear relationship between, here is a claim that you have made on your website, in your IFU, in your other documents. Remember, claims can live in a lot of different places within your ecosystem. And the notified body pays attention to all of those claims, and that they're going to want to see if you use, words like better, faster, stronger, right? That this our stent is better than, or is more efficacious in this particular type of population. Let's say something like that. They're going to want to see somewhere that there's data that directly supports the claim that your device performs this way and I'm using an example of a certain patient population. So that means that the data that you're going to need to present to support that claim, it has to be documented that it is in the claim, the data come from that patient population, and that it is for the indications on the IFU of that device. And this is the last part of it. What kind of data? Sarah talked about weighting and appraisal, level of evidence. All data sets presented in a CER, a PER, a scientific validity report, whatever the document is, all data sets need to be independently weighted and appraised for their value of their contribution. Data sets that are weak in their they're maybe the type of data that's just anecdotal or verbal, or it's not a part of a clinical trial. That's a weaker level, or a less valuable data set than obviously the gold standard RCT data sets. So weighting and appraisal, familiarize yourself internally with the best weighting and appraisal tools to use, and every data set has to be weighted and appraised. And then that allows the notified body to make their own evaluation and judgment on whether or not the clinical evidence is enough to support a clinical claim. the level of the evidence, and that whatever you say in the claim is being supported by clinical data.
Yeah, and this is a good place to point out one of the questions that just came in. It was focused on what is the difference between intended purpose and indications in terms of this data sufficiency for the evidence. So the easiest way to think about it is that the intended purpose is sort of that overarching, when, where, and why would your device be indicated. The indication is more specifics in terms of those PICO terms. So intended purpose encompasses the population, the indications, the comparators, and the outcomes. It's the big picture, whereas the indications is just why. The why would you be using this, or where would you be using this device? So an indication, for instance, if you're talking about like a vascular stent, it could be like, are you indicated for coronary artery versus peripheral artery disease? That would be the indications. The intended purpose would be, broader than that. It would incorporate aspects of the populations for whom you would be appropriate, as well as the indications. So if you are devising an intended purpose with multiple indications, you have to have evidence to support each claim standalone independently. You can't just say, well, most of my evidence is in the peripheral vascular system, but sure, you could use me in the coronary artery. If you don't have that evidence, you can't make that jump. So that's where you may want to go back and say, either I need to do a study because I'm really dead set on keeping this extra indication, or right now, I'm just going to get it approved based on what I can prove and what the evidence supports right now.
Thanks. Thank you, Lori and Sarah. I'm going to go ahead and on to the second question. This is also from registration. Can you please discuss the new MDCG 2023-7? Sarah, I know you put these notes in here, so I'll go ahead and hand this to you this time.
Yes. So I'm glad someone brought this up, because this really speaks to some of the challenges and some of the frustrations that folks have had as far as legacy devices or wet devices. There originally, there was this assumption that clinical data on the subject device is required, not from equivalent devices. But this guidance that just got published in December states very clearly that any clinical data that falls within the definition of clinical data, including equivalence data, is acceptable. And it also speaks to that it defines when contracts are or are not required to claim equivalence. So this helps you better understand if you can use a clinical data from another manufacturer's device to support the scientific validity or the state of the art for your device. So it really is going to be extremely helpful for making that clarification and making people feel like they're on sound footing as far as using a very, very similar device from a competitor in order to support your device claims.
Sarah,
So
I want to jump in because I want to make sure that it's very clear to everybody what you just said, which is, are you saying that with this new MDCG guidance, that no longer is a requirement that there has to be a contract between company X and company Y, who are not related to each other, but are you saying that sufficient levels of access, whatever that means, is replacing that? We need to see a signed contract so that each company can they can share their technical file? What does that mean exactly? Is that what you're
Yeah, this one, I would definitely refer them to reading through this document. It's got a lot of very specific details, but what it does say is that data from another manufacturer's equivalent device can be used without a contract. And that would fall under this exemption case 4 that they document, where the manufacturer has a contract with it at least one manufacturer. So it does open up some other potential routes for collecting data if you
very
fall,
specific.
but it's very specific. It is, but it actually provides us additional details over what had been published previously, where we were just making assumptions, or, you know, it does give you a lot more flexibility, I think, in terms of what the expectations are. And so I think it could be very useful for those legacy or the wet devices.
Yeah, very late breaking news there. So great.
Yeah.
Thank you.
Excellent. All right, I'm going to go to the next question. What is the difference in data presentation between MDR device and legacy MDR device? Laura, you want to jump in first on this one?
So difference in data presentation, I'm going to frame that within a CER for under MDR. Let's just talk about that. So difference in data presentation in a CER between an MDR device and a legacy MDR device. None. There's no difference. Really, there's no get out of jail free card if you're a legacy or a wet technology device. It is the rules apply to all equally. With regards to presentation, you walk through your CER template. Let's talk a little bit about the CER template, and that, you know, Criterion Edge has developed a pressure tested CER template since 2017, when we first started doing this work, and it has evolved multiple times as we learn more. We are all learning more as we go along. And the short story from our opinion is that there's no difference in the CER template itself and how you march through the story from beginning to end, and what the through line is throughout that very complex document. The through line of what is the story you're trying to tell, and where are you taking the notified body? You know, what's the end of the story? So all of that, it doesn't matter whether you're talking about a novel device, a brand new device that's being submitted for the first time for CE mark, or it's a legacy MDR device. It has to be, the story has to be coherent, the data have to be clear, all data sets have to be analyzed, appraised, and weighted, and related back to the claims, the IFU, the intended purpose, patient population of the device, no matter if it's legacy or brand new.
Excellent. Thank you, Lori. Before I go to our last question for registration and start to get to live questions, I want to remind you all that you can submit any questions via the Q&A button in the webinar tools bar. I also want to remind you that you can reach out to us at consult, that's CONSULT at criterionedge.com, to schedule some time to talk with our specialist about any of these topics. We're available to chat with you about your current or upcoming project needs and answer more specific questions. So with that, I'm going to go ahead into the next question. Is clinical performance evaluation of biobanked samples enough to obtain approval? I think that's a specific one. Sarah, I think you have these notes as well, so I'll hand that to you.
Yeah. This is again coming from that MedTech Europe, and this came from a question submitted earlier. But there's actually a question in the Q&A right now that's really similar, and it's asking for a novel IVDR device. What are the benefits of moving from a retrospective evaluation of biobank samples to prospective sample collection studies? You know, and I totally agree. There are benefits and risks of both strategies, and ultimately all of the regulations and all of the guidance is telling us that you can demonstrate clinical performance directly or indirectly, or use a combination thereof. So it really will depend on your device. So let's say you have, if this is from an IVDR perspective and you're talking about a rare disease, biobanking may be your best bet, because if you're trying to set up a prospective study for a very rare disease, it could be years before that study is going to get enough enrolled to actually be clinically relevant. The challenge, though, is that you need to make sure that you have enough of the data and that the samples have been collected, and that the collection process meets all of the same rules and regulations as a typical clinical study. So you may not necessarily have to have the the informed consent if it's been de-identified, but that's something that you need to look into going into the process. But ultimately, it really could save you a heck of a lot of time. But if you think about it, like that direct demonstration is the data that are based on your particular device produced by that manufacturer, and they're obtained from studies prospectively or from biobanked leftover samples. Whereas indirect is going to be more data based on your literature searches, published information regarding your device, or a comparison with another reference device. So those method comparison papers. Direct is always going to be a stronger evidence as far as clinical performance data compared to that literature search alone or the method comparison studies alone. So it's a balance, but you can absolutely rely on the biobanked samples, and in some cases that is going to save you quite a bit of time and energy by taking advantage of samples that have already been obtained.
All right, thank you, Sarah. We have another question here from the audience. What is the typical time frame for state-of-the-art data, and which time frame is typically applied for device-related data? This is device. Lori, why don't you take a shot at this one?
Sure. A typical time frame for state-of-the-art data. Let's just talk about time frames in general. The rule, essentially, in whether you're writing a PER or a CPR over in the IVD world, or you're writing a CER in the medical device world. Let's talk for a minute about data time ranges. You're going to have multiple sources of data, your PMS data, data from published literature, manufacturer-held data, typically clinical trial data, and certainly PMS data is manufacturer-held, and so is PMCF data, are all manufacturer-held. All of those are typical sources of data on the device. Now we're talking about the state-of-the-art. What's the state-of-the-art search frame, and why they all need to be the same? Because when you start the state-of-the-art analysis, the goal of that analysis is setting the stage for where does your device under evaluation fit within the milieu of the state-of-the-art of its indications and patient population, what it's indicated for, and what does that state-of-the-art world look like? What kind of data? What is happening out there? What is expected for a device that is, opens a coronary stent? What is expected in the framework of state-of-the-art from all data related to all coronary stents? So the time frame of your state-of-the-art search to build your state-of-the-art conversation or section has to be the same as the rest of the data sets that you're going to be pulling, your manufacturer held with. And so what is that, generally speaking? It's generally you're looking back 1 or 2 years. Now, I will say in state-of-the-art, when you're looking more at guidelines and other types of things, you can go further back. What you shouldn't do is be shorter than your time frames for your clinical data. So essentially, all the time frames should match. Certainly for the data going to support the clinical data, going to support your device under evaluation. The state-of-the-art, remember too, that when you're doing a state-of-the-art search, you're setting, again, the landscape, and there's always a third search in a CER, and that's your competitor device search. Now that the competitor device search most definitely should have the same date range as your subject device searches, because you're going to derive out of your competitor device search your acceptance criteria for your safety and performance objective. Those objective criteria, those acceptance criteria come directly from your competitor search. So don't forget the competitor search as well. Time frames should all match.
Yeah, this actually reminds me of an example that we just encountered. As we're writing different SVRs and trying to establish scientific validity for an in vitro device, a lot of times you'll notice these international standards that are being set out. And that comes from your state-of-the-art literature search and your guideline search. Suppose you have a changeover where the international standard has been updated, and let's say that happened 4 years ago. In that case, if you are aligned with the new current international standard, you really wouldn't want to go back further because you're comparing the accuracy of your test using an outdated standard. So it's really a good example of how that state-of-the-art search helps you kind of inform and drive the decision making as far as refining your performance objectives, your time frame, your choice of competitors, making sure that they all make sense and that they're aligned. Because if you throw in older data, you're going to have a skewed data set where they're going to say you're comparing old standards to new standards. There's been a change. And how are you distinguishing between devices that use the new versus the old?
Sarah, you're absolutely right there. And thanks for that additional clarity. I can tell the folks on the call that are listening is that in our experience, working with many, many clients and writing now hundreds of MDR, CERs, and many documents in the IVD space as well, and having reviewed notified body responses and helping our clients answer them. They will go, the notified body will go to your data first. That is, that's like red meat to them. They will go to it every single time and go over it with a fine-tooth comb. The comments that Sarah just made, that I just made, all of that. If they find anything that's assailable that they don't like, it will get written up. Because think about it, the data prove or disprove if your device is safe and performs as intended. It's all about the data. And if there's something that they question about the data, where it came from, the data sources, the data time frame, mismatches, that's what they'll hang their hat on first every single time. If you don't pay attention to anything else in your CER, or pay attention to your data, it has to be scrupulous. And that includes, by the way, your searches. Sarah and I have lots of opinions about searches, and they will start there. Is that search, the search that you do is on the internet, or it's not under best systematic literature review, best practices, that you're dead in the water before you start, because they're questioning the data itself in a search that is not compliant with best practices. So those are our thoughts.
All right, let's go to our next question. We got them coming in hot and heavy here, so we'll keep going. How can acceptability criteria be defined when there's more than one approach for treatment? For example, device or surgical technique. Sarah, you want to tackle this one?
Yeah, so this is a really good example too of sort of the difference between like the approach you would take for a novel device or versus a device where you have direct competitors. So the acceptability criteria are basically the scale by which you're going to measure your own device's performance. So you want it to be as close to your device as possible. So if you have a device that has direct competitors on the market, you're going to say they perform the sensitivity of other very similar competitor devices is 95 to 100%. If you're comparing yourself to something like a totally different diagnostic technique that, like, let's say, it has a totally different level of diagnostic sensitivity. So like, for instance, again, with the COVID testing, if you're a PCR test and you're comparing yourself to a chest x-ray, it's apples and oranges. You're clearly going to be better than a chest x-ray. So if you're hanging your hat on, well, I'm better than a chest x-ray, and you're ignoring your competitor data, they're not going to be happy with that. But if you are the first PCR test for COVID out the door and the only other alternative out there is chest X-ray, then absolutely you scream that in your like data presentation. You say, look, compared to the other alternative, which is all we've got, I'm offering a novel approach that is far more accurate, has far more clinical benefit to the patients. That's why I'm state of the art. I currently am standing alone in the field being state of the art, but I'm still state of the art compared to the other alternatives. So think about it as, if you are dead set on doing your treatment or testing in one particular way and you can pick something similar, that's a competitor. If you are the only option in that entire type of treatment or type of testing, then you have to rely on an alternative in order to devise your acceptability criteria.
And I'll just jump in here too to just say from giving an example in the medical device world is a stent versus a cabbage, surgical revascularization. Excuse me. A stent, you just can't. It would not be valid for you to present in a CER acceptability criteria for, say, safety for a stent placement, which is your device is a stent, and compare that to the safety data from a coronary revascularization. Those are, again, apples and oranges. So if the question here is, how can acceptability criteria be defined when there's more than one approach for treatment? Fine. You choose the approach that is relevant to your device. So again, a stent has nothing to do with coronary revascularization. So you would only use acceptability criteria related to stents. So that, I hope that helps. It's pretty straightforward. Again, as Sarah said, apples and oranges. You can't compare one to the other. Sometimes when we're talking about state of the art, what we say to people is, for example, a baseball team. Okay. So if your baseball team is a high school baseball team, are they playing the same game as the New York Yankees? Absolutely. Do they have the same people on the same positions, number of men on the people on the field? Yes. All the rules are the same. Yes. But can you compare, excuse me, I've got something in my throat today. Can you compare the Yankees to the performance of your local high school? Of course not. So you compare the high school team to other high school teams. You compare the Yankees to the Boston Red Sox. And that's how you, so keep that in mind too. That when you're comparing apples to apples, it's also the level of, who you're comparing it to.
Yeah, and one more question that actually just came up in the live Q&A is defining acceptability criteria from the SOTA already in the CEP. How do you do that if the search is planned for the CER? It sort of gets into this concept of the loop. And just keep in mind, this is a process. And the idea is that they want to see you document your process. You're starting with the state of the art. So you're starting with, what do I know now? Here's in my plan. As you start to write your CER, you're saying, here's how I'm going to seek to identify new information, or if there is a new potential emerging use for my device that's off-label, or if there's a new complication for my device that's being talked about, an interference or a cross-reactivity for an IVD, that's something you've got to have a way that you can objectively identify that information moving forward. So that's the planning part of it. You want the planning to come in to say, I have a way that helps me be assured that I'm going to identify this information moving forward. The CER is, today, as I'm writing this right now, here's what I know. So the CER is more like a snapshot in time, whereas the plan is a plan moving forward to make sure that you're going to keep catching all of that relevant data moving forward.
Yeah, well said.
All right.
Yeah, sorry, jumped the gun on that one, but I thought,
Yeah,
oh, that's a good one.
I got onto the slide
Yeah.
eventually. Okay, we got another one coming from the audience. Sarah, I think this one's for you. If a device has been in the market for years and there are similar devices also, but as treatment is not included in medical guidelines, is it considered a novel device or can be stated as SOTA if there is clinical evidence available?
This is where it gets tricky. Like if you don't have your device being explicitly called out in the treatment guidelines, you would have to use that, the basic background information and the results of preclinical studies to say this is potentially a new emerging. Like it's not in the guidelines yet, but the guidelines take a minute to catch up to how it's being used in the real world. So as you're looking at like what's being used, how is the field evolving, don't think that just because you're not in the guidelines right now, that doesn't mean that you're not state of the art. So it just means that that's where it's really important to show that the maturity of the technology, sort of how the field is emerging, how this treatment is emerging, and how it's evolving as like this entire landscape. So that would be my main advice, is that the guidelines are sometimes slow to catch up because they're based on a large data set, and so you just have to show here's the data. It's just it hasn't been incorporated into the recommendations as of yet.
All right, we got another one coming from the audience. I think this one's for you, Lori. When a medical device's CR update can stop, sorry, when, I think it means when can a medical device's CR update stop if the device's MDD certificate expires? Does the CR have to be updated until the expiration date of the last batch of MDD device or once the MDD certificate gets expired?
I'm going to be careful answering this one. I did see the question come in, and I wanted to, raise my hand, here internally amongst all of us, as I can take that. But I'm afraid that we're not regulatory affairs experts. I can say that we haven't done, and Sarah, correct me if I'm wrong, I'm not sure that we've ever written a CER after the certificate has expired. So
I can't remember one. No.
Not at all clear, nor would I want to quote regulations about how, and I understand the question. The question is, okay, the certificate is expired. How long do we have to do the CER thing before we're off the hook, is how I sort of see this. I think there are questions like, are you still selling your device, for example? Mm-hmm. Is it still on the shelves everywhere and people, it can still be implanted in patients? Again, not a regulatory affairs expert on this, but those two questions, if either one of them are yes, I think you still have to continue evaluating the device. It's out there in the market. It's still being implanted. And then there's questions about how long do you track a device that's been implanted? And in other words, even if it's not being sold anymore, stent X isn't being sold anymore. Is it still in people? And how long do you track the safety and performance of stent X, even though it's no longer on the market? It's still in people. And it's within the scope of how long that device is useful or its lifetime. And those are questions that I would get very clear answers on before stopping writing your clinical evaluations on a yearly or semi-annual. That's a question for your notified body.
Perfect. Our next question, and we're getting low on time here, but again, if you have any questions, feel free to put them into the Q&A section. If we don't get to them today, we'll get back to you on them. But this one, can retrospective data be used instead of prospective data for clinical efficacy when developing AIAMD for rare diseases? How do these principles apply to SAMDs? Sarah, you want to grab this one?
Yes, yes. So this is an exciting aspect that I think we're going to be tackling in our next webinar. Artificial intelligence as a medical device is really being introduced into a lot of different types of clinical pathways, both on the IVD side and the MDR side. So, it can be used for diagnosis and screening, for prognosis, supportive treatment. It can be used to anticipate changes or predict changes in blood glucose for continuous glucose monitoring. It can be used to evaluate your risk for developing a new condition based on your test results. So we're seeing it pop up all over. When you're dealing with a rare disease, retrospective data is always going to be your friend, because you would have to take so long to collect enough prospective data. It's not to say that you shouldn't try to embark on a prospective study, but just recognize that the results of that study are going to take you quite a bit of time to accumulate compared to using biobanked samples or analyzing readily available retrospective data. That's still part of your clinical assessment and that evidence. And a lot of these same principles are going to apply to software as a medical device. It's sometimes tricky to separate the medical devices from sort of what we typically think of as the software. But software can be in a medical device. It can be part of a component of an in vitro device. We're seeing it over and over again, and there's been some recent publications that have really helped us better understand how to approach it from the same basic perspective, just taking into account that you're dealing with software, not a specific device. So we will be doing our very next webinar on software as a medical device and do more of a deep dive into that topic, because it's a little too big to try to tackle it in one Q&A session here.
Excellent. Yeah, we've come to the end of our time. So I'm going to go ahead and flip. We got through most questions, so good job, guys. And thank you, audience, for your excellent questions. This was productive as usual. All right.
And so we will definitely incorporate that AI as a medical device and software as a medical device in this next upcoming webinar here. So feel free to register for that webinar and send us any additional questions, and we will do some digging before we meet again.
Yeah, so our team will be sharing some links in the chat, and we'll remain on the webinar for a few more minutes so you can access these. If you enjoyed our webinar today, you may want to sign up to be notified about additional upcoming webinars produced by us. You can do so by clicking the link provided in the chat. If you still have questions about the content we discussed today, if you'd like to learn more about our services, you can schedule a free appointment with our specialists by contacting us again at consult, that's CONSULT at criterionedge.com, or you can book a meeting at a time for you at the link that you'll see in the chat. The webinar is recorded, will be available soon to watch on our website or sent out to you an email with the slides and all these announcements. When you exit the webinar, please take a short survey and let us know how you enjoyed today's talk and what other topics you'd like us to cover in the future. Thank you, everyone, for attending and for all your questions. We hope this presentation gave you more insights into your work, and I hope you have a great rest of your day, and we'll see you next time. Thank you.